Hello, my name is Kelly Pablo, and I am a UNE online medical physiology student. Today's date is November 5th, 2019, and I'm here to complete the reproductive system portfolio. So let's begin. So for the reproductive portfolio, I wanted to talk about feedback loops in a non-gravid female reproductive system. We can't start talking about the regulation of the female reproductive system without first going over the importance of hormones. Hormones influence almost every organ in the system in the body and are critically important to maintaining homeostasis. They are chemical messengers released by endocrine tissue directly into the bloodstream. From here, they can circulate throughout the body and reach even the most distant cells. However, they only target cells with an appropriate receptor. There are three main types of hormones, and they're classified by their composition. Steroids, like the estradiol molecule depicted right here, are derived from cholesterol. Peptide and amine hormones are protein-based. Depending on the type and its cellular target, a hormone can cause several different effects. For example, the lipid-soluble steroids can cross the plasma membrane and influence gene expression to regulate the production of proteins. The more polar peptide and amine hormones tend to bind receptors on the cell membrane and trigger a signaling cascade that causes changes within the cell. When a hormone induces the secretion or inhibition of other hormones, it is considered a trophic hormone. Many of the endocrine pathways in the body rely on this multi-step process, as it can be controlled more precisely. When the end products of a hormonal signal stimulate additional release of that hormone, positive feedback occurs. When the end products inhibit release of their initial hormone, the reverse happens. This negative feedback is much more prevalent and allows endocrine pathways to regulate themselves. The female reproductive cycle, more commonly known as the menstrual cycle, primarily involves changes in both the ovaries and the uterus, preparing the tissues for possible pregnancy. However, there are two organs which play an important role in regulating these changes: the hypothalamus and the anterior pituitary. Let's start with the hypothalamus, a small structure in the base of the brain that functions as a bridge between the nervous and endocrine systems. It secretes gonadotropin-releasing hormone, or GnRH, which then activates cells in the anterior pituitary, the body's master endocrine gland. These cells then produce both follicle-stimulating hormone, FSH, and luteinizing hormone, LH, which travel through the bloodstream to the ovaries. Here, they trigger a complex series of events, which we will cover in the next few slides. For now, just note that they stimulate the release of several ovarian hormones: estrogen, progesterone, inhibin, and anti-Mullerian hormone, abbreviated as AMH. These hormones generally apply negative feedback on the hypothalamus and pituitary, inhibiting further secretions. In addition to their trophic effects, they also induce changes in the uterus, namely to the inner lining known as the endometrium. As mentioned before, the hypothalamus regulates the menstrual cycle by producing gonadotropin-releasing hormone. Without the interference of other hormones, these secretions actually occur in a series of pulses, releasing every 1 to 3 hours. These pulses are echoed by the anterior pituitary and act as a sort of metronome, helping time the stages of the menstrual cycle. While the precise mechanisms are still a mystery, scientists believe that neural signals from the rest of the brain, circadian rhythms, and a hormone known as kisspeptin all contribute to the function of this endocrine clock. The release of GnRH is also controlled by both positive and negative feedback from ovarian hormones. When it reaches the anterior pituitary, GnRH stimulates the release of both follicle-stimulating hormone and luteinizing hormone. While both of these travel to the ovary, they each target a different cell type. FSH is important for waking the dormant follicles and stimulates the granulosa tissue, causing a limited number of them to start maturing. LH also stimulates follicular growth, but does so by targeting thecal cells, resulting in increased estrogen production. A surge of luteinizing hormone also acts as a trigger for ovulation, when the mature egg is released from the follicle and swept into the fallopian tube. Within the ovary are a number of follicles, oocytes surrounded by a layer of granulosa cells. Primordial follicles form during fetal development, and some of them slowly enlarge into primary follicles. During puberty, a portion of these primary follicles develop an external theca, becoming secondary follicles. A still smaller number of these continue growing into tertiary follicles, characterized by the beginnings of a fluid-filled antrum. Every month, only one of these tertiary follicles completes its maturation during the follicular phase of the ovarian cycle. This final maturation process is triggered by follicle-stimulating hormone and luteinizing hormone released from the anterior pituitary. FSH works on granulosa cells, causing them to divide and secrete three important hormones. Anti-Mullerian hormone desensitizes other follicles to FSH, while inhibin exerts negative feedback on the pituitary to decrease FSH levels in the blood. Together, they limit the number of tertiary follicles undergoing development, preventing too many from being activated at one time. Progesterone is also secreted and stimulates the further production of both the GnRH from the hypothalamus and luteinizing hormone from the pituitary. Luteinizing hormone, on the other hand, acts on thecal cells, causing them to grow and divide. They also produce androgens, which diffuse into neighboring granulosa and are modified into estrogen by the enzyme aromatase. Estrogen is the dominant hormone of the follicular phase and can elicit a variety of effects depending on its concentration and target location. First, it stimulates the uterine lining to begin proliferating. Then, at low to moderate levels, it inhibits GnRH and by extension FSH and LH. Simultaneously, it creates a positive feedback loop in granulosa cells by stimulating them to produce even more estrogen. As a result, estrogen levels eventually peak, and at these higher concentrations have the opposite effect on the hypothalamus and anterior pituitary, resulting in a massive surge of luteinizing hormone. There is also a smaller surge of FSH, but its release is restricted by the action of inhibin. This flood of LH prompts the release of the oocyte from the dominant follicle, signaling the end of the follicular phase. After the follicle has ruptured, the thecal cells fold into the antrum and mix with granulosa. These cells combine to form the corpus luteum, a hormone-secreting structure that remains in the ovary. Its formation marks the beginning of the luteal phase. The corpus luteum secretes three hormones: progesterone, estrogen, and inhibin. Progesterone, like estrogen, has a dual effect on the hypothalamus and anterior pituitary. It stimulates them at low concentrations and inhibits them at higher concentrations. The combination of high progesterone, moderate estrogen, and inhibin virtually shuts down the production of GnRH, FSH, and LH. Although all three hormones contribute to the events of the luteal phase, progesterone is the major player. It furthers the development of the endometrial lining, causing it to store lipids and glycogen while also secreting mucus. All preparations for possible embryo implantation. Interestingly, it also raises the basal body temperature. The corpus luteum usually lasts about 10 to 12 days. If no fertilization occurs, it degrades into an inert structure known as the corpus albicans. Progesterone, estrogen, and inhibin levels fall, which results in the removal of negative feedback for GnRH, FSH, and LH. Once progesterone is lost, the endometrial lining also loses its blood supply and eventually dies. The dead tissue sloughs off and is passed out the vagina during menses. Finally, gonadotropin levels return to normal and the cycle begins again. This diagram from the Silverthorne text is an excellent depiction of the menstrual cycle, showing the overlap between both the ovarian and uterine cycles. It begins with the onset of menstruation, after the corpus luteum has degraded and the gonadotropin levels return to normal. While menses is occurring, rising FSH levels recruit a clutch of tertiary follicles to begin maturing. As they develop, there is a steady buildup of estrogen, which inhibits GnRH, FSH, and LH up to a point. It also stimulates the endometrium to begin proliferating. When estrogen levels peak, its feedback on the hypothalamus and pituitary switches from negative to positive, resulting in a massive surge of luteinizing hormone. As you can see, FSH levels also peak,
but to a lesser extent due to rising levels of inhibin. This follicular phase lasts anywhere from 10 to 21 days. The LH surge then triggers one dominant egg to burst from its follicle and begins the start of the luteal and secretory phases. The now empty follicle transforms into the corpus luteum, which takes over hormone production in the ovary. The combination of high levels of progesterone and inhibin coupled with moderate levels of estrogen virtually silence FSH and LH production. Progesterone also stimulates the endometrium to complete its preparations for embryo and implantation, becoming more vascularized, storing more nutrients, and secreting mucus. After approximately 12 days, the corpus luteum involutes into an inactive form. Deprived of progesterone, the thickened endometrium dies and sloughs off during menses. The return of normal GnRH, FSH, and LH levels then restarts the cycle. The whole process takes about 4 weeks to complete. As you can see, hormonal control of the female reproductive system is quite complex. Many stages are regulated by both negative and positive feedback simultaneously. In addition, estrogen and progesterone can either stimulate or inhibit gonadotropin production, depending on their concentrations. In order to simplify these feedback loops, this slide breaks down the various trophic effects of the ovarian hormones. At low to moderate levels, estrogen inhibits GnRH, FSH, and LH. It also stimulates its own production by granulosa cells. When these levels peak towards the end of the follicular phase, the control switches to positive feedback, stimulating GnRH and prompting a surge of luteinizing hormone. FSH would also surge, but it is partially blocked by inhibin. Progesterone has the opposite effect of estrogen. At low levels, it stimulates GnRH and LH, cooperating with the increased estrogen to initiate the LH surge. At high levels, it inhibits gonadotropin production. Coupled with inhibin and moderate levels of estrogen, this shuts down the LH and FSH during the luteal phase. When inhibition is removed, gonadotropin levels return to normal. Inhibin and anti-Müllerian hormone are the easiest to understand, as they only inhibit the function of FSH. Hopefully, this makes the interactions between these hormones a little clearer. And finally, this brings us to the end of the lecture. Here's a list of the references that I used to prepare it. Thank you for listening.